Okay, welcome everybody to the first session of the day, where we'll get to learn all about invasive terrestrial plants. Your moderator for this session will be Nicole, who is a communications coordinator at the ISC. So she's one of the people behind all of the social media, newsletters, media scans, things like that. So thanks so much, Nicole, and you can take it away.
Thank you, Maddie. Hi, everyone. I'm looking forward to moderating this session. We have an amazing lineup of speakers sharing their research and knowledge about invasive terrestrial plants. I'd like to introduce today's first speaker. We have Vicki Zhang and Peter Kotanen of the University of Toronto, and they'll be discussing barriers to plant invasions at the edge of the Arctic. Thank you so much for kicking off the first session of the day, and yeah, whenever you're ready.
Awesome. Thanks so much, Nicole. Can everyone hear me? Peter, can you hear me?
Yeah.
Okay, good. Awesome. Hi, everyone. My name is Vicki, and I'm joined today with my supervisor, Peter. So I'll be doing the presentation, and then we're happy to take questions at the end. And we'll be presenting some of the work coming out of our lab looking at plant invasions in the Canadian Arctic. So I'm going to situate us first and talk about the fact that at high latitudes, such as in the Arctic and Antarctic biomes, there are very few non-native plants. A recent review looking at latitudinal trends of latitudinal patterns of species distributions found that the species richness of non-native species decreases towards the poles, as I'm showing here, and this is on a log scale, so every unit is a tenfold decrease. And in addition, the proportion of non-native species is highest at mid-latitudes and always decreases near the equator as well as at high latitudes. And this low proportion of non-native species might be driven by two different processes. So for example, near the equator, this could be likely due to biotic resistance and competition, as the tropics are very highly diverse. But at higher latitudes, such as in the Arctic and Antarctic, the lower proportions of non-native species might be due to some other reasons, such as abiotic factors and propagular pressure. The Arctic is one of the few areas in the world that is minimally affected so far by non-native species, but they're slowly becoming an emerging problem now. And many recent studies have documented the presence of non-native species in the Arctic. So here I'm showing you just vascular plants found in the Arctic Circle, but this also includes aquatic and other terrestrial non-native species as well. In Canada, we see a latitudinal pattern of non-native species emerge. So in temperate regions such as where I currently am in Toronto or at U of T's field station at KSR, there's a very high percentage of non-native species. And as we move upward and northward, there are fewer non-native species taking up these northern biomes, such as in the boreal forest and northern temperate regions. The outlier to this very nice regression is Churchill, Manitoba, where over 20% of the flora are non-native species. But before I dive into this anomaly, let's first think about some of the barriers that a species might face as it is invading an area and before it is considered invasive. So a non-native species needs to first move past a biogeographic barrier into a new environment. And once they're in this new environment, they might be faced with abiotic factors and environmental factors that are not initially ideal and that they have to adapt to. After that, a species might face reproductive barriers where they aren't able to reproduce or produce viable seed and instead might persist vegetatively or clonally. There are also barriers to dispersal, so a species might be unable to physically move past its first initial point of introduction. And it's only when a species has moved past all of these barriers that it might be considered widespread and dominant. And on a continental scale in Canada, it's presumable that what is really limiting non-native species here is climate or environmental barriers. Since most invaders originate from temperate regions, they don't do as well in the colder areas. But at the local scale, such as in Churchill, Manitoba, these barriers might be a little more complicated. And to date, there are very few experimental work done on invasions in the Canadian North. Let me introduce you to Churchill, Manitoba. This is a very small town of about 900 inhabitants, and it lies on the coast of the Hudson Bay, right at the edge of the boreal forest tree line. It's also the traditional meeting place of the Inuit, the Dene, the Swampy Cree, and the Métis nations, and is also popularly known as the polar bear capital of the world. Importantly for us, though, Churchill has been a site of a lot of activity. So it was a Hudson Bay Company post since 1717. It was a former military base, and it also has a grain port and a railway link to the south. And so this area here is what I consider ground zero for invasions. It's been heavily bombarded by non-native species, where anything that wasn't grain was simply thrown out into the surrounding areas and into the filling up the local dumps and around buildings. However, almost all of these non-native species that have bombarded Churchill have been restricted to the town footprint. Very few have escaped these human disturbed areas, even though they've been here for over 60 years. So here I'm showing you the surrounding tundra and boreal forest, and they remain very natural and almost free of non-native species. And this is very different from temperate North America and other temperate regions where non-native species can be found in almost all ecosystems. So our lab studies factors that might promote the persistence of non-native species or limit their spread as a barrier in these northern environments. So we conduct fieldwork on the ground in Churchill using many non-native model species, including some familiar ones that you might recognize here, such as common toadflax, common plantain, and common dandelion. And today's talk will focus on some experimental results that came from testing several environmental barriers, as well as the dispersal ability of some non-native species. But I'm happy to also talk about some reproductive observations or reproductive barriers that we've observed through doing this work as well. Let's start with looking at how dispersal abilities might be limiting non-native species in Churchill. As you may know, seeds of non-native species might be unable to disperse into new areas without physical introduction, and once they're present, they might fail at that seedling stage, and this can result in localized invasions. As well, roads can serve as a corridor for invasion, especially via vehicles and human footwear. And for unpaved roads, which is common in Churchill, with roadside maintenance or recreational vehicles, we can have long distance dispersal of non-native seeds. And these dispersal limitations might also explain why there are fewer non-native species at higher latitudes. There's just limited accessibility, resulting in lower introduction events and lower propagule pressure. So we transplanted non-native species onto roadsides in both the boreal and the tundra and boreal forest ecosystems, and we monitored their survival and performance over 2 years after transplant. And if they're able to survive in these novel areas, then there's evidence that dispersal might be actually limiting them from getting there. But once they're there, they are able to establish. Here I'm showing you survival along the y-axis across 2 years post-transplant. So I transplanted these in 2021. And then the points are colored by the species, and the shapes indicate the habitat where the transplants were moved to. So let me first point you to the very significant and drastic drop in mortality by the 2nd year post-transplant. For instance, dandelion in purple dropped to 50% 1 year post-transplant, and this year the mortality was 100%. So there's just no more transplants of dandelions in the tundra plots. And similarly, for common plantain in orange and toadflax in green, the mortality was really significantly high, and much higher than the drop in mortality in the boreal forest. So in the boreal forest, mortality was much lower, and there's really high survival, at least for linaria and plantain. And so after these 2 years of observations, we found that plants are able to survive after translocation into new sites in the boreal forest, indicating that they are dispersal limited to some extent. After I move them, they're able to survive in the boreal forest. And this also matches broad biogeographical trends in Canada, where the boreal forest is much more easily invaded and has a higher proportion of non-native species compared to the tundra, perhaps because it is further from the bay, slightly further south, a little more sheltered. Next up, let's look at the effects of warming. So a colder temperature in the tundra might be a barrier for non-native species, limiting them from further spread. In warmer microsites, it's been observed that non-native species can do better, which is one reason why temperate regions are much more heavily invaded. The Canadian Arctic is warming at a rate that is 3 to 4 times the average rate of warming globally. And with this warmer climate means that there is a greater potential for non-native species to invade. Currently, native species are dominating this environment as they've had long periods of time to adapt. But with the warming temperatures, that might create more suitable habitats and niches for non-native species. So to test the effect of warming on these non-native species, I transplanted them and added their seeds into these mini greenhouse exclosures. And I asked whether transplants and seeds do better in this simulated warmer environment. Here I'm showing you transplant survival along the primary y-axis and seed germination along the secondary y-axis, separated by species. And again, the data is colored by treatment. And so far, after 1 year of observations, there is maybe a trend that plants transplanted into the warming treatments do better, but that's currently not significant. But there is a marginally significant difference in seed germination, at least for dandelion. So dandelion germinants were much greater in warming plots compared to the control plots. And I should say for these seeds, I added seeds on the order of hundreds. And so the germination rate is fairly low. It's about 10%, but it's not 0. So this indicates that a warmer temperature can increase seed production and it may increase survival, but we do need more data here. Finally, let's talk about buildings as a suitable microhabitat. So this work is done by a lab mate, Francine, who just defended her thesis, and she was looking at the effect of buildings and anthropogenic structures that might improve microsites. So some sides of buildings, such as the south and west sides, can trap heat, and certain exposures might also trap water and provide a shelter and a windbreak for non-native species. And so Francine asked whether there were certain exposures that might be warmer, such as the south or west sides, and whether that will increase the abundance and fitness of common dandelion. And she answered this by conducting surveys around 22 buildings in Churchill. Because of the orientation of the Churchill town site, all the buildings were angled in a northwest, northeast, southwest, southeast exposure, and that's what I'm coloring here. And I'm showing you the average abundance of dandelions in varying distance from 0 meters to 5 meters away from the buildings. There was a higher abundance of dandelions on the southern sides of buildings, so the darker bars indicate southeast and southwest abundances, and the northeast and northwest orientations had lower abundances consistently. And in addition, there were more dandelions closer to buildings, and abundances decreased at 4 to 5 meters away from buildings, indicating that this sheltering effect of buildings decreases with increasing distance. And there's also evidence that a closer proximity to buildings and southern facing exposures can increase population abundances of dandelion. And I don't show it here, but we also did collect soil surface temperature data, and we found that temperatures were warmer closer to buildings and on southern exposures. Next, let's look at flowering, which we use as a proxy for fitness and how that differed across exposures. And this was done with the help of an undergrad, Brianna. We found that the highest proportion of flowering individuals occurred on the southwest sides of buildings, and then the lowest proportion of flowering occurred on the northwest sides. And so this is also evidence that southern exposures can increase fitness, and in this case, increasing flowering of dandelions. This work is still novel, it's preliminary, and it will continue in future years, but it's really key to understanding the emerging issue of invasions in the Canadian North. Just to summarize, high latitude environments are currently dominated by native vegetation, and non-native species are failing to spread. And this might be due to several barriers. For instance, low dispersal ability is limiting spread into suitable habitats in the boreal forest, and colder temperatures are really limiting growth and survival and germination. and these both lead to failed invasions so far. But some species, such as dandelion, might benefit from anthropogenic changes that can lead to higher plant performance and fitness. There's evidence that some non-native species are going to benefit from these barriers, benefit if these barriers to invasions are removed. So for example, if there are anthropogenic factors that can increase vectors for increase long distance dispersal by acting as vectors, or with anthropogenic changes such as buildings or other human-made structures. Climate and human activity and anthropogenic change can make northern ecosystems much more vulnerable to invasion. And so here we're seeing that Churchill could really be a hotspot for future invasions, which can threaten the native tundra species that are already there and that have important ecological and cultural significance. I'd like to thank my lab for doing a lot of this work, as well as our funders and our other supporters out in the field. And I'm happy to take any questions with Peter.
Thank you so much, Vicki. It's very interesting about the implications of invasives in the tundra and climate change and other factors. So yes, if anyone has questions for Vicki and Dr. Kotanen, you can put Q&A box. Okay. We have the first question. How did you, someone asked, how did you ensure that the introduced species would not spread?
That's a really, really good question. And I get that all the time. So we are very, very careful with all of our transplants and seeds. Everything is marked not in 1 or 2, but in 3 different ways. So we can always find them again. And we're always ensuring that we're able to remove them at the end of the experiment. As well, their current lack of spread is indicative that they can't spread, or they're just dying. And so we are pretty sure that if we transplant them out there, they will die in a couple years, and that's what we're seeing. But again, we are taking basically every precaution that we can. We're marking them out, we're able to find them, we're monitoring them multiple times a day all summer long. And so we think that there's very little risk of them actually spreading, and we don't want to be the first ones to spread them in Churchill, for sure.
Thank you so much. Couple more minutes for questions. If anyone else has any questions, you can type them in the Q&A box. Okay. Next question, is there any evidence that the soil biome is affected by this introduction?
That's a really good question. We've looked at it the other way around. We looked at whether the soil biome is affecting introduction. And to that, we found that not really. The soil might have some effect, but it's not significant. It depends on other abiotic factors. Whether it's the other way around, such as invasive species coming in and modifying the soil, I can't say that for certain here in Churchill, but we do see evidence of that elsewhere. And I don't know if Peter has anything to add to that.
No, we haven't really looked at this in detail. It's an interesting thought. We've wondered whether there might be novel mycorrhizae in particular arriving, but we don't have evidence of it at this point.
Great. Okay, we have time for one more question. Are there dandelion flowers in introduced areas? If so, how did you prevent them blowing off and spreading?
Yeah, I'm assuming by introduced areas here, you mean into the sites that we transplanted them, so into the boreal and tundra. We are checking them daily, so we are monitoring everything from growth to flowering to seed production. So we know which ones are about to flower and about to produce seeds. And then when they are about to, or when they are producing seeds, we collect them. The plan initially was to collect their seeds and look at seed count and fitness, but only 1, maybe 2 have produced flowers and seeds. And so we collected them, and we're just making sure that they're not blowing around.
Great. Thank you so much, Vicki and Peter, for joining us today, sharing your talk. If anyone else has any more questions for Vicki and Peter, Vicki, you can answer them in the Q&A box. There's an option after you accept questions to type an answer. And you can also leave your contact info if you wish for any further questions after. Thank you so much for joining us.
Thanks so much.
Okay, next up, we have Spencer Karau of the University of Ottawa. He is discussing key functional plant traits to predict the rate of colonization of invasive plants in North America. Thank you for joining us, Spencer.
Yeah, it's a pleasure to be here. Can everyone hear me just fine? And can everyone see my presentation?
Yeah, perfect. Your presentation looks good to me.
Great. All right, I'll jump right into it. So hello, everyone. My name is Spencer. I'm a master's student at the University of Ottawa, and today I wanted to talk to you a bit about the research I'm doing for my thesis. So my thesis is entitled Using Biological Traits to Predict the Rate of Colonization of Invasive Plants in North America. So why is this topic something that is important? Well, we have a lot of research in the literature that studies the impacts of invasive plants on the ranges that they invade. And there's also a lot of research that looks at what factors really go into facilitating the rate that these plants spread. But what we don't have is sort of an agreement on which factors are the most important. And there's really a lack of clarity on what in particular facilitates spread. Additionally, there's no reason to suggest that invasions aren't something that's going to keep occurring. And what's more is that with factors like climate change, it's possible that invasions are going to be occurring in places where in the past they never would have had the chance to occur. So getting a better understanding of how these plants spread in novel ranges and what actually facilitates this spread is a good idea for helping managers when they have to deal with these invasive plants. So what's the question we set out to answer? We wanted to know which, if any, biological traits can be used to predict the rate at which invasive plant species will colonize newly invaded ranges. So to answer this question, we need to sort of set up a variety of moving parts. So I'm going to try and give a bit of a background on everything, and then we'll jump into the results. So the first part we should take a look at is, what are biological traits? When we talk about biological traits, we're talking about the intrinsic characteristics of the plants themselves. These are basically the features of the plant that make the plant the way it is, or the species. And we want to know how these traits relate to spread rate. So for our study, we looked at 3 traits. The first trait we looked at was seed dry mass. Typically, plants with a heavier seed are more resistant to tough conditions, and they start off with more resources. So this can make them more tolerant of unfavorable conditions. But species with heavier seeds are also typically harder to disperse. So with that in mind, we have sort of 2 hypotheses, and we're going to make 2 hypotheses about each of our traits, and we'll explain what we mean by the terms in the hypotheses a bit later. But for now, we predicted that species with a higher average seed mass will reach climate equilibrium faster, but will have lower maximum colonization rates. The second trait we looked at was plant height, just how tall is the plant at its maximum height. And we thought that, or it's known in literature that plants that are taller typically can outcompete those that are shorter for light and other resources. So with that in mind, we thought that plants with a higher maximum height will reach climate equilibrium faster, but typically takes longer to grow larger. So we hypothesized that we predicted that plants that have that are taller will have lower maximum colonization rates. And finally, we looked at specific leaf area. This is just the ratio of the leaf's area to its mass for a species. And typically high specific leaf areas are related to high relative growth rates. And with that in mind, we predicted that species with high specific leaf areas will reach climate equilibrium faster and have higher maximum colonization rates. Okay, so we're talking a lot about spread, but how do invasive plants usually spread? Well, they typically follow what's known as an invasion curve, and this is just a way to look at how these species propagate once they arrive in a novel range, and it can be broken down into several stages. The first thing that happens is transport, whether it's brought over on the bottom of a boat or brought over for ornamental purposes and escapes. A species somehow makes its way from its native range to North America. After transport, a colonization period begins where the species has to establish itself in the novel range and build a self-sustaining population. Once this has occurred, the plant can begin spreading across the landscape. And this is typically where the maximum rate of colonization is observed. And finally, after the plant has had time to spread and it's sort of reached the extent of where it's going to be in the novel range, we say that the plant has sort of reached equilibrium. It's no longer increasing its distribution and it's no longer decreasing its distribution. So typically when we think about invasion curves, we think about them in geographic space, but we can also think about them in climate space. And this is important for us because when we look at the rate of colonization of our species in our analysis, we look at it in both geographic space and climate space. So quickly, what's the difference between geographic space and climate space? Well, geographic space is simply put, you can think of it as where plants or species are found on a map. So in this example, you could say that the species is in the northeast quadrant of the map, or the plants are in 25% of the range. And if these plants were to spread across this map, proportionally, the geographic space would increase as well. Meanwhile, climate space can be thought of as what environmental conditions are the plants found in. So if you considered each of these different colored squares on our map to be a different climate based on how warm and how wet it was, we could split this range up into 5 different climate spaces. And if we see that our plant is in 2 of those 5 climate spaces, we could say the plants occupy 40% of the available climate space. So with that being in mind, we can see how even though the plants are in the same spots in reality, the amount of geographic space and the amount of climate space they inhabit can be different. In addition to traits, we also included some other factors in our analysis. First factor was native range size. We included this in models that were dealing with expansion of species in geographic space, and it's just how big was the plant's range in its native range. The second factor we included was growth form. This is just a categorical variable. Is the species a tree? Is the species herbaceous? Is it a climber? Is it a shrub? And finally, we also had native niche breadth. This is just a measure of what sort of climates or niches was the species found in its native range. Was it found in a broad range of niches, or was it found in very specialized niches? And we use this covariate in our models that we're dealing with expansion in climate space. Another thing to note is that when we ran models that used plant height, we actually removed the growth form term. And this was because plant height and growth form were seen to be significantly correlated with one another. So where is all this data coming from? Well, our occurrence data is coming from the Global Biodiversity Information Facility. Our climate data comes from WorldClim, and our trait data is coming from TRI, the global database of plant traits. These are all excellent open source databases that are curated and have data submitted to them all the time. And without the generous submissions of researchers, this type of analysis wouldn't be possible. So for my study, we looked at 258 species that were identified by the Center for Agriculture and Biodiversity to be invasive to North America. These species came from a variety of different native ranges and were from a variety of growth forms, albeit the majority of which were herbaceous. So I collected and cleaned data for all 258 of these species, and I was able to find data for 247 of them. And of these 247, 105 of them reached equilibrium in climate space. Okay, so we have our biological traits that we want to look at. We have our other factors in our analysis. And we begin answering the question of, did traits influence colonization rate? So the first response variable we're going to use to answer this question is time to equilibrium. This is just the amount of years that it took for a species to reach equilibrium in climate space since its introduction in North America. So if a plant was introduced in 1850 and it took until 1950 to reach climate equilibrium, time to equilibrium would be 100 years. And we calculated this for every species that reached equilibrium in climate space. And you can see that on the y-axis of our plots here, that the times to equilibriums for our species range from 25 years to 125 years. On the x-axis, you can see our traits, and note that these x-axes have been logged. And what you'll see is that there's a lot of scatter here. Our points aren't really conforming to the line of best fit. And it doesn't seem like there's a great correlation between our response variable and our trait variables. What I'm showing you here are the individual terms that go into each of these models. So for example, our top model the model would be comprised of seed mass, a native niche breadth, and growth form. And when we put these terms together to make a prediction about how long it took a species to reach equilibrium in climate space, so the time to equilibrium, we can see that the adjusted R-squared for that model is quite low. And it's quite low for all of our models dealing with time to equilibrium. What's more, we found that adding the trait terms to our model actually did not improve the predictive power of the model in all cases except for with plant height. So that is to say, when we added seed dry mass or specific leaf area to our model with our other covariates, the predictive power did not measurably change. Okay, so the second response variable we used to try to answer our question was looking at rate of spread, specifically the maximum rate of spread in climate space over a 30-year period. So to calculate this response variable, we plotted our occupied climate space over time at 5-year intervals, and we did this for every species. We calculated every possible 30-year slope, and we isolated for the highest slope. And doing that, we were able to get a value for every species. And we wanted to look at the data in terms of rates because only 105 of our species were able to reach climate equilibrium. So by looking at rates, we were able to leverage all 247 species into our analysis. Doing this, you'll see our response variable once again on the Y and our trait variables on the X, and note again that the traits have been logged. We see that there's again a good deal of scatter, and it doesn't seem like our trait variables are well correlated with our response variable. This is echoed in that the adjusted R-squared values for our models are again quite low. And what we noticed here was that actually none of our trait values serve to increase the predictive power of our models. So adding the traits in did not improve the predictive power of our models in any of these cases. So finally, we had one more response variable that we were going to look at. This was looking at the same thing as our second response variable, but rather looking at spread rate in climate space, we were looking at spread rate in geographic space. So it's the exact same calculation as before, but looking at things in terms of geographic space. And what we found was that when we had our final set of response variables, we had to log it to normalize our data. We wanted to look at the data in geographic space for two main reasons. Firstly, most of the current literature is focused on expansion in geographic space. So by doing this, we can more readily compare our results to what's out there in the literature. And secondly, the sort of barriers to expansion might be different in geographic space and climate space, and likely are different. And some traits might prove to be more important to spread in one space over the other. So by looking at things in both geographic space and climate space, we can start to look into that. Doing this, we get probably our best correlations between our response variables and our trait variables, although they are still not very strongly correlated. And note again that the trait axes have been logged. And what we'll see is that while our adjusted R-squared values for these models are highest that we have seen, they are still quite low. Note that because we're dealing with geographic space here, our native niche breadth term has been replaced with a native range size term. And similarly to what we saw with time to equilibrium, the only trait that actually improves or makes a measurable difference on the predictive power of our models is plant height. Adding seed dry mass or specific leaf area to their respective models did not serve to increase the predictive power. So what does this mean? What can we say? What can we garner from these figures? Well, of the three traits we looked at, the only one that really seemed to make an impact on predictive power of our response variable was plant height. And it was only able to do that for 2 of our 3 response variables. Moreover, when it did make an impact, the overall adjusted R-squared value of the model was still quite low, with the highest one being less than 0.2. Meanwhile, our other factors, our covariates in our analysis, seem to be significant predictors the majority of the time, but as we saw from our adjusted R-squared values, the overall predictive power of these terms was still quite low. We also noticed that in most instances, adding the trait terms to our models did not seem to increase or decrease or make a measurable impact on the predictive power of our models. So this suggests that whatever limited predictive power we are getting is coming from these non-trait factors, from these covariates. So what can we garner from all of this information? Well, it seems that the traits that we looked at, our study traits, do not well predict spread rates. at large scales or in general contexts. So when we calculated our species averages, we considered species from research that was performed in their native range and in the invaded range. And we aggregated all that data to create species averages. And doing that, it seems like it was hard to sort of see any correlation between a trait and a response variable that was tied to colonization rate. The other factors in our analysis, like native range size and native niche breadth, seem to play a greater role, but as we saw with our adjusted R-squared values, these roles still seem to be relatively limited. So all this, along with information in the literature, seems to suggest that perhaps at smaller scales or when we have a higher data resolution and we know more about the specific context of the environment that's going to be invaded, we could say more about the predictive power of a trait, or a certain biological trait might be more predictive. But when we look at things at this wide scale and we are sort of grouping these varied landscapes together, it's really difficult for us to say anything about the predictive power of traits. So thank you very much for your attention, everyone. This work would not have been possible without the following people, and I'm really happy to take any questions.
Thank you so much, Spencer, for your presentation. We have a few questions, so if anyone could direct your questions to the Q&A box, and we can answer from there. While we're waiting for, I'm just wondering, I'm sure a lot of viewers here understand what it is, but it's kind of a basic question. Would you be able to explain to me like what climate equilibrium means exactly? That's probably a very broad question, but
Yeah, it's a tough distinction to make, and it's one that we sort of, it's hard to explain. So when we say climate equilibrium, we're basically talking about the point in time, or the point at which a species has encountered all of its all the climate spaces that it's going to encounter. So if you look at this example, you can see that this range has divided into 5 climates. So when a species arrives, it's going to enter into the first climate space that it's going to encounter. And as it spreads, it's going to encounter more and more climate spaces. So over time, the species will continue spreading and continue encountering different climate spaces, but eventually it's going to stop spreading, and it's not going to increase the amount of climate spaces it enters, and it's not going to decrease the amount of climate spaces it enters, theoretically. And that's the period of equilibrium. So it's not approaching more climate space, and it's not retracting from climate space. So the amount of climate space that it occupies is at equilibrium.
Perfect. Thank you. That makes sense. Thank you for that explanation, Spencer. And we do have a question in the chat. If you were to do additional analysis to answer the question to predict which species will be invasive in the future, what variables might you investigate further?
So that's a good question. We're kind of limited in the variables that we could select, in that we are dealing with open access data. So we have to sort of choose from what we have. So let me just read the question.
It would be in the answered box.
Yeah. So I'd really like to do some more analysis on traits related to, species. Yeah, so variables. So I'd really like to do some more analysis into variables related to dispersion. So sort of what dispersal method are these plants using, and how far do they typically disperse? And I think that would be a good trait to look at, and would speak a lot to the rate at which a plant spreads, and also the time that it takes for them to reach climate equilibrium.
Thank you, Spencer. So there's time is up, but there is one more question. Wouldn't reproductive traits be more predictive? I'm wondering if you could answer that in the Q&A box chat. And also you can leave your email if you want to for any further questions.
Sounds
thank
good.
you so much, Spencer. That's super interesting.
Thank you very much.
Thank you. Okay. So next up, we have Hayden Wilson from the Kawartha Land Trust. She'll be discussing dense grasses as a dog strangling vine deterrent. Hi, Hayden. Thanks for joining us.
Hello, everybody. Okay, hope everybody can see that slide deck all right. Computer's a little bit slow, so I apologize if there's a bit of lag. But really cool presentations so far. It's been really fun to listen into what's going on, and happy to talk a little bit about what we've been doing at the Land Trust to try to manage dog strangling vine. As I'm sure many of you folks know who do some kind of land management, or at least in areas in southern and central Ontario, dog strangling vine is a pretty aggressive invasive species. I won't get too much into the life history of DSV and kind of its range on the landscape. But if you have DSV in your ranges, I'm sure you're probably sick of hearing about it anyway. So I thought I'd bring you a bit of a good news story to look at two different case studies of two different methods of dog strangling vine management that we've employed on one of our properties. They're both sort of focused around using grasses in different species as a way to sort of create a physical and ecological barrier for DSV invasion. As many people know, DSV spreads really rapidly. It's carried by the wind. The rhizal system spreads incredibly fast. So different ways to get around that are what I'm mainly going to be talking about today. I won't be able to give sort of an exact, here's how you beat DSV on your property, but here at least is what has worked for us in a couple of our different managed areas. So as a quick little primer, my name is Hayden Wilson, Land Stewardship Manager at the Kawartha Land Trust. We're a land conservation charity that focuses on Peterborough and the Kawarthas on protecting and conserving land in this area. One of those properties that we do a lot of management on is the Biotyping Trails property, which is the one I'll be talking about today. So the two different ecosystems that we've sort of developed in a sense to help manage dog strangling vine on the landscape are tall grass prairie ecosystems, which are not particularly common, especially in Ontario. They sort of have a wider, more extended range into the States. We do have small remnant populations of them and a lot of historical evidence to point to a lot of southern Ontario at one point being covered with a pretty extensive range of sort of patchy savanna, open tall grass prairie, open woodland, oak woodland areas. So tall grass prairies are really denoted by their tall grasses, many of which can grow over 8 feet high and often are populated thinly with some variety of fire-resistant trees. These are fire-dependent ecosystems, so they do receive regular burns either naturally or through man-made practices to maintain that opening. So this is an image of tallgrass prairie at Biotyping Trails, and it stands pretty well against dog strangling vine. I'll get into the details of how we do that in a minute, but basically everywhere around that you're not seeing where the tall grasses are in frame, DSV is more or less the dominant plant. It is everywhere on this property. And when I say it is dominant, it is the dominant plant. It is everywhere in those classic wide swaths and stands that it tends to spread out into. The other ecosystem that we manage for dog strangling vine prevention is one that is entirely sort of, in a sense, made up. It's a very unique ecosystem that consists of almost entirely, and this might be frustrating for some people to hear, almost entirely black locust as a tree species, as the canopy, and then a mixture of different fescue grasses as the understory. Now, of course, is this a, quote unquote native ecosystem? Not really. The bounds of that can be stretched and pulled depending on how you define native and non-native and what's useful and what isn't on the landscape. But this ecosystem is available to us due to property's history of once having a plantation of black locust on there and around an existing or an old farmhouse from the original, from the settlers who came to that property in that area. So this sort of stand of black locust already existed. And it provided us this sort of interesting and weird opportunity to get in there and do some management against DSV. As you can see in the background here around these two people, there is almost nothing else in the understory aside from these low grasses, and the black locust is pretty much the entire canopy. So it's quite almost park-like in a way. As you're walking through, it's almost like a managed park area. And it is also incredibly good at managing dog strangling vine. So how do we actually do that? Well, we have to sort of look at things in the context of where this property actually is. So this is the Valley of Trails property, which exists in sort of the Pontypool area. So this is just a little bit south of Lindsay, Ontario, for those of you who know where that is, just a little bit north of the Ganaraska Forest. And if you're still not sure where that is, it's about 30, 40 minutes north of Oshawa. So this property is sort of set in this area that otherwise historically likely would have been mostly dominated by prairie or oak savanna sort of ecosystems. It's very sandy, it's right on the edge of the Oak Ridges Moraine. So there's sort of an interesting characteristic to this site already. And unfortunately, a plethora of dog strangling vine in the entire area as well. So again, we're certainly going to be focusing on these two different areas, looking at dog strangling vine management in the prairie and dog strangling vine and black locust management in this black locust stand. So the main real driver of both of these methods, one of them being sort of focused on native grasses and one of them being focused on non-native grasses, is looking at occupying as much soil space as you possibly can. For anybody who's ever done any dog strangling vine management or knows anything about this plant, and I'll flip back a slide here, the root masses that are in the photo on the left here are incredibly dense. The more you cut them, the more they prune them, the more they spread, and you just literally physically take up every inch of soil that's available to them. The root systems are relatively shallow depending on the soil system that they're in, but still they really want to occupy as much soil space as possible. So our answer to that is to occupy more soil space than dog strangling vine if we possibly can. We go about that by using sort of these two different mixtures of grasses. So one is sort of more on the far left side, what you'd see as sort of lawn grass, really short root systems that spread out somewhat fibrously and kind of cover really wide areas physically above ground. The other one is using native grasses, specifically native tall grasses, which many of these species in this interesting little diagram are, that have these incredibly dense root systems that reach really far down into the ground. They're really well adapted to these drought prone conditions and are excellent at maintaining themselves in these really kind of areas that are open and really easily accessible for invasion by invasive species like dog strangling vine, which just frankly don't care what kind of soil they grow in. They'll go anywhere. There's some habitats that grow better or worse in, but this area is particularly good for them. There's almost no competition. So really the main thing that is important for both of these methods, both the black locusts and understory grasses and tall grasses, is site preparation. So this is absolutely the most important part of doing any of this work that we have found over the years, is really nailing it down and getting excellent site preparation done as much as is possible. So for something like the black locust, I'll talk a little bit about what that looks like in a minute, but to jump into the prairie management of things, and again, I'm sort of going to be glossing over a lot of this stuff. If you want to talk about more specific how to make a prairie, let me know. Happy to chat about that. But really, site preparation in both of these is key. And that includes getting as much sort of area cleared and open and treated as much as you possibly can to prevent dog strangling vine reinvasion really easily as you've opened up these areas. For doing management in the prairie, that might look like doing a prescribed burn to open these areas up. It might look like manually tilling things up and removing a lot of DSV out of the soil from that way, or using classically herbicides to treat an area and then put things back in. In the locust stand and out in the prairie as well, it also looks like removing a lot of what I like to call catch areas. So DSV in particular is a species that is wind distributed for the most part. They have sort of milkweed-like seeds that spread out on the wind, and they often catch in areas where there are brush piles or, you know, really heavy amounts of thatch from old grasses and things like that. And they land on those piles and they get into them, and then you can never get them back out, because no matter how much you spray or pull or do anything, they're still protected by this cover. So removing as much of the cover from the surface as you possibly can really is one of the best ways to go about doing site preparation. So you can get into these areas that are really heavily infested, do your treatment following the best management practices, and then go forward with this technique. So some of the species of grasses that we use to reestablish the prairie, I'll go really quickly over these, are sort of four main species. One is yellow prairie grass, yellow savanna grass, goes by a bunch of different names. This one image here. Oh, sorry, moving forward. There we go. Okay, the next one would be little blue stem, a much sort of denser, lower grass that tends to spread out and clump a little bit. All of these grasses that I'm talking about as well are sort of bunch grasses, so you can imagine them kind of growing in little islands together. The next species is big blue stem or turkeyfoot, a massive, massive grass growing over 6 to 7 feet tall, and again, really occupying a lot of soil space. Has a really aggressive growth pattern too. It will spread really rapidly. And the fourth one being switchgrass as another species that tends to have a really wide seed dispersal. You can see these seed heads are really widespread. So as the switchgrass grows, it tends to expand out quite quickly and cover more and more area. So I often get asked, oh, sorry, and then there's other species like Canada wild rye, which can be a really good establishment species. And that kind of leads into a question I often get with these is, well, if you're doing this DSV management and you're, using herbicides to get things up of the soil and making this space open and available, why don't you try and go with maximum diversity right out of the gate? Try and, really establish a really healthy, functioning ecosystem rather than just focusing on grasses. Well, it would be really nice to be able to put things in other than these tall grasses you can see in these plug trays here. Species like wild bergamot or butterfly milkweed or sweet everlasting. Maybe even species at risk like dense blazing star, or one that spreads really rapidly like gray-headed coneflower. Those would be great to put in, but they're just not tough enough. And that's something we've learned over time doing this, is that we really have to establish a physical barrier before we can establish a biological one. And these grasses are acting as that physical barrier for DSV reinvasion, again, through the density of the root masses and through just having something in the soil that isn't dog strangling vine or another invasive species or another species that tends to thrive in disturbed environments. So as much as I'd like to say, yeah, you can go in and make your prairie in 1 year, it's just not going to happen. You have to take this really slowly and one step at a time. So looking towards those grasses, the actual method of us putting them in there, if you're starting, you have your hypothetical blank slate field, you're starting in this really sort of area that probably had a lot of dog strangling vine invasion, potentially even other invasive species like white sweet clover, maybe chickweed, maybe some other things like that. They just aren't your target species. Really, what's most important for a lot of these is having excellent ground contact and exposure. To do that, we go about putting them in the ground, these tall grasses, in slightly different ways. We've often done in the past where we've just, again, tilled up, used herbicide or some other kind of site preparation technique to just open things up. And then we could, potentially cast seed out in those areas. We could leave those areas open for invasion through natural processes. But one of the best ways that we've found is to collect these tall grass seeds and then grow them as plugs. You can see that they grow pretty dense, and the photo on the right here shows just after these plugs were only growing for maybe 2 months, they're already starting to not really root bound themselves in their cells, but really aggressively putting roots into the ground. And that's one of the reasons, again, why we chose or why tall grasses are so effective at this, is that they spend their first year just absolutely pumping roots into the ground as much as they can. So to do that, we take these tall grasses, we plunk them into the soil in bare spaces that exist within the prairie, that we've done those sort of management techniques on. And in a few years, potentially even 1 or 2, they really fill that space out quite rapidly and prevent dog strangling vine from getting soil contact and then being able to spread and spread. There we go. Oh, skip one. So that also does look like continued maintenance over time. Over many years, we have volunteers head out and do management on these different properties, including pulling out dog strangling vine that does make it into the prairie and other management techniques to get rid of them, including using graboid-based herbicides to just target or to not target dog strangling vine in the prairie. So this isn't something that is, you can set it and forget it. Prairies need maintenance. And again, there's a lot more to prairie maintenance and establishment than what I'm talking about here, but it is incredibly effective at keeping dog strangling vine from coming in and spreading out really widely over these big extensive swaths. So the other method that we use in the black locust stand is a little bit more simple and perhaps a little bit more of a primer that kind of gets to the essence of what this sort of strategy looks like. And that's using something like a fescue grass mix. Now, obviously, as you're sort of looking through some of these mixes, you can see, do they contain a massive amount of native species? No, they do not. But they do contain species that are relatively easy to manage, that are low maintenance, and that if you wanted to get rid of them, you can get rid of them pretty easily. So we use this as sort of a physical barrier to prevent dog strangling from coming into this ecosystem. Does that mean I would go into a beautiful deciduous forest that is full of trout lilies and hepatica and everything and dump this all over the place? Probably not. This is sort of a technique that's used for areas that are already incredibly overrun by invasive species and don't have a lot of native species still left in the area. So this is sort of creating a clean slate as it might be to start with. So there are a couple different mixes that we use. One is from Wildflower Farms in Coldwater, Ontario. The other one is sort of a bigger bulk mix that you can buy from DLF Pig Seed. They both have species that really spread widely and cover soil surface area. So again, instead of the prairie grasses, which are a lower sort of occupying more of the subsurface space, these are physically carpeting the entire surface and not letting any contact happen. Again, this technique really is sort of focused on having good site prep really built into this. So again, we're removing a lot of the downwoody debris. We're scraping old grasses away. We're removing any DSV that exists on site to try and get as much open area as we possibly can for distribution of these grasses. We don't tend to grow plugs for these grasses. They don't really like seeming to grow in trays. They really prefer it if you just sort of scrape the soil and toss them on. They're really low maintenance, they're really easy to use. And the nice thing is that they're really easy to sort of expand out on site. The prairie management we've been doing for over a decade now, as well as managing this black locust stand. But one of the main things we've learned from both of these techniques is to bite these off in bite-sized chunks. The more edges that you have, the more territory you have to defend. So being able to start this in sort of an area you can just see, just in this image right here, looking where these people are sort of spread out. This is roughly maybe a 30-meter square area, so it's not massive, but doing this in these really concentrated areas, maximally occupying soil space and above soil space has really helped to make sure that reinvasion doesn't happen in the same way.
Stop that. It's been a little over 15 minutes, so there's 3 minutes left for questions.
For sure, thank you. And I'm just about to wrap up here. Thanks for the heads up. So again, does this mean we never have to go in and do any management? No, of course not. There's always things that we have to do. We have to go in and pull DSV and manage it where there are these catch areas where it tends to hold on to. And then there's also things like the grasses will sort of naturally succeed out and sort of crowd themselves out. That opens things up into doing other planting opportunities, like adding in understory diversity through shrub plantings in these areas are sort of succeeding out and dying out, and doing management of the locust stand itself by thinning out the canopy, making more gaps, and then introducing diversity from there. And it leads to really quite an aesthetically pleasing area as well, which I think is a huge benefit of this. So that is my presentation. Thank you, everybody, for listening. Sorry, I went a little bit over time there, but I just get excited about prairies. I'll stop sharing and take any questions.
Thank you. So there's a couple questions in the chat. First one, what time of year are you doing the site prep in the woodland areas?
What time of year? Mostly it's focused on either early in the spring or late in the fall, kind of when a lot of things have gone dormant and it's just easier to do management. You can really do it about any time of year, though. It doesn't seem to really make a huge difference. This seed mix that we're using, you can spread down basically at any time, but it does do better either being cast out in the spring when it's really wet or cast out just before the winter. So it's really plastic and easy depending on what you want to do. You can do it multiple times per year.
Great, thank you. And one more, how do you keep birds from eating all the seeds, e.g.
How do we keep, sorry, what?
How do you keep birds from eating all the seeds, such as with Ecolon?
Oh yeah, excellent question. You have to kind of buy a little bit more than you think you're going to need. They will come in and try and eat it, but The benefit of this site is that there's a lot of natural forage for birds around. So it's not so much that we're casting it out on the front lawn and all the starlings are coming down and eating it. There's a lot of natural forage for them, but they still do come down and eat it. That being said, they also still come down and eat all the prairie seed that we scatter out. So we try and time the seed scattering to not coincide with maximum amounts of birds being around. That being said, it is difficult to keep them from eating everything.
For sure. Okay, great. Thank you so much, Hayden. If you want to, there is actually one more question, but if you could answer that in the chat or the Q&A box. I might also have a question for Eagle on that might come in the chat, and you can also share your email in the chat, and that goes for all panelists. Thank you so much for giving us your talk, Hayden. That's super interesting to learn about what's going on at Kawartha Land Trust. Thank you so much.
Awesome, thank you.
Okay, so next up we have Dreisilker from the Morton Arboretum talking about public gardens as sentinels against invasive plants.
Thank you, Nicole. Can you see my screen okay? Can you hear me?
Yep, looks great, and I can hear you well. Thank you so much, Kurt.
Great. Well, thank you so much for having me. Thank you, everybody, for joining. As Nicole mentioned, my name is Kurt Dreisilker. I'm with the Morton Arboretum. We are just outside of Chicago, Illinois, down in the United States. And today I'm going to talk about what public gardens, botanic gardens and arboreta are doing to help shed light on plant invasions. So really kind of defining or redefining our role in the plant invasion process here. See if my technology works okay. Just a bit about the Morton Arboretum in general. As a tree museum, we have over 222,000 living plants within our collections. And so these come from all over the world. You can see some of the stats down on the bottom of the screen there from 42 different countries. And we have lots of threatened plants in our collections of trees and shrubs as well. So this is a very important part of our mission. This is essentially what we do. But we've also been, questioned about having all these non-native plants, and you can obviously see the connection with plant invasions. So we're over 100 years old. Joy Morton founded the Morton Arboretum in 1922. You can see a picture of his lovely estate and a picture of him planting a tree back then. And in the first few years of our existence, many trees, hundreds of thousands of trees and shrubs were planted across the landscape. And this is just an image from back then, looking across the entire Arboretum, where you can see lots of introduced plants and trees growing all across the landscape. So this is kind of a setting for introducing plants into a context that, into a landscape context before we even knew about plant invasions. Here's a report from a different botanic garden in the United States, reporting kind of an unusual situation that happened within their own garden. It's about this plant called Celastris orbiculatus. It was known by a different name back then. But you can see in kind of the second paragraph here, in 1897, the bittersweet was introduced. Within a few years, large tangled masses of bittersweet appeared on shrubbery and rocks nearby. The casual observer expressed surprise at the abundant growth of the shrubby bittersweet in the garden. And jumping down to the next box, the color scheme was appreciated by the birds. And it's also noted that they're not slow in taking advantage of the food supply and consequently scattering the seeds which start new colonies of the vine where they may rest after a time of feeding. So you can see the process begins. So the idea here is that botanic gardens and arboreta across the continent have this unusual view about plant behavior. And so we introduce plants into our collections, and over time we may be finding them spreading around within the property that we manage. So this is back in 1922 when it was introduced. Now here's the population of this species today. Obviously it has spread quite wide and far across the eastern United States and into Canada. This population is not just the responsibility of that one garden. Obviously this particular plant has a much longer story, which helps to contribute to its plant invasion. And at the Morton Arboretum, we've certainly had our battles with this plant as well. You can see some images where the vine has grown into some of the tree canopy across our property. And you can see the severed limbs, severed vines, where the plant has now been desiccating when I took these images a number of years ago. So this plant has become invasive. Now, we recognize it as an invasive species. Here's just another example of a plant that most of us are probably familiar with, Rhamnus cathartica. You can see the wide expanse of its population across the continent. Similar story. However, now in the Chicago Region Trees Initiative Tree Census report for the Chicago region, European buckthorn occupies 36% of the tree canopy in the region. So here's another example of a plant that was introduced, many years ago before we knew about plant invasions, and now we can see the impact that it's having on our urban forests. So it's not a secret, it's not unknown to people that horticulture is a pathway to plant invasion. And here's some data from Teresa Cully's paper out of the University of Cincinnati. Out of 295 species listed as invasive in the Midwestern United States, many of them have their ties to ornamental horticulture. So you can see some statistics here in the charts. 89% of woody plants have ties to ornamental horticulture, as well as 65% of vines and 31% of the herbaceous plants. All have ties to horticulture. So this is really more complex than just what botanic gardens and arboreta do. This also involves industry, of course. So I'm not going to belabor the point about this invasion curve. I suspect that most of us on the call today have a really good grasp of what this is, what this means. However, I just want to make the point that most of us are probably familiar with dealing with the plant populations on the right side of the curve. So in the red, where the plant is infesting large areas, the plant has been around for a long time, and it will take a lot of money and resources to control that. The initiative I'm talking about today is focusing where the blue arrow is on the left, where the plants are just introduced into a botanic garden or arboretum context, and we can just start to see when plants start spreading from cultivation. So this initiative is now called Public Gardens as Sentinels Against Invasive Plants. This initiative started back in 2016 when a bunch of gardens gathered together at Morton Arboretum to talk through what is happening within our various gardens. What do we think about that? Is it problematic? And so on and so forth. So we had lots of questions over the course of 2 days. And of course, we did not answer any of them, or all of them, I should say. It really prompted the whole initiative called Public Gardens as Sentinels Against Invasive Plants. So we did recognize, however, that gardens are actually perfectly poised and positioned as sentinels because we have curated collections of plants that come from other parts of the world, as well as the native range where the garden is located. We have natural areas oftentimes where we can find plants spreading into those uncultivated areas. Professional staff, curators and horticulturalists who dedicate their time to managing the landscape. And we have lots of information as well. So we have databases and labels on the trees and lots of information about those plants. So if we all pooled together and understood what was happening with these plants as they escaped from cultivation, we could have this vast network of gardens and botanic gardens and arboretum showing larger scale trends. And so this map just shows locations of many of the gardens across the North America. So in 2018, we officially formed the working group, and we've been meeting monthly ever since, including through today, the present time. So we did some foundation building first. We had to understand exactly what was happening. What were people thinking across the garden industry? We found through some survey work that 91% of the gardens have actually observed plants escaping from cultivation. 89% of the gardens control those plants when they are spreading from cultivation. And then 89% of the gardens thought it would also be useful to understand what other gardens are dealing with, what other species, and how are they dealing with those escaped populations. Despite this information, at that time, no platform existed to share this information. It was really up to just individuals to reach out to our peers and find out what was happening within the rest of the industry. So by 2019, we set about developing standards for the gardens to follow. And so we did this. Here's the URL on the bottom left, and then an email address that you can send messages to if you have questions or have any further comments on. So I'll leave those on the bottom of the screen you can see there. But here's just a couple images of those guidelines in the image on the left. You can download those from the website. And then probably the most importantly, we also have developed standard categories or ranks where gardens can all talk the same language, characterizing what those plant populations are actually doing equally. So we all have kind of the same language that we're using to categorize these. And then we also have recommended garden policies for how to deal with these plants as they kind of climb through the ranks of plant spread or population spread. So once we have standards developed, gardens can contribute their data to the database, which we've developed here as well. So this database is actually by registration only. Gardens, through significant feedback from the garden industry, this registration only process was really important because gardens wanted to network within the garden industry, understanding what was happening within their peer properties across the continent. And then we also thought it was important for people to know what's happening with this industry as well. What the data is showing on a wider basis. So getting the information out of the public garden sector was also important, and I'll talk about that in a second. But we went about in 2021, really, once we had these two tools developed, the standards and the database, we went about recruiting gardens to participate. And so many gardens actually stepped in and actively contributed their data. By 2022 and 23, we invited other stakeholders to the conversation where we thought it was important to get the information out. So gardens had been pooling this data, contributing their data to the database. But as I mentioned, that data was kind of locked behind that registration wall. And so we know that in order for this initiative to help prevent plants from becoming invasive, we really needed to communicate this information to other stakeholders, like landscape architects, like propagators, the green industry, nurseries, invasive plant councils, and so on. So we invited this group of organizations to the Morton Arboretum back in November of 2022 to talk through just how to communicate that. What do all these stakeholder groups need in order to effectively communicate this information to their stakeholders? And so we got this idea as a result of that conversation to develop an alert. This alert system is a PDF that I'll show in a second, but it really just contributes or communicates larger scale trends to these stakeholders. So if you are interested in the results, I will show you some sort of real-time data. You can log into our website and see this data dashboard that's almost live. This is actually from the end of January, a screenshot from that. But we have 38 gardens participating across the United States and into Canada. Across all 38 gardens, they've contributed 887 records of plants escaping from cultivation. You can see on the bottom left here, you can see a pie chart where those different categories of invasive, potentially invasive, watch list and assessed as invasive are broken down into the pie chart there. You can see in the map on the top right, you can see where the states, well, you can see where the gardens are coming from, represented by states. Note that the garden names are not shown in this image. And then perhaps where most people want to see is the bottom right, where we have this bar chart showing the number of gardens recording various taxa. And so you can see the winner at this point, if you will, is Philodendron amirense, which is the Amur cork tree. And you can scroll across to see some of the other taxa that are being listed most commonly. So
just a heads up, sorry, that you have 2 more minutes left. Thank you.
Thank you. Appreciate that. So as I mentioned, Philodendron amurense is the amarcord tree. 11 gardens are actually reporting this particular species. At the Morton Arboretum, we have a significant history with this particular species. We've ranked it potentially invasive, and I'm willing to share that as a representative from the Morton Arboretum. However, we have 10 additional gardens reporting across all the other categories as well. So you can see the breakdown is invasive, potentially invasive, watch list, and assessed as invasive. You can see the numbers of gardens reporting per category rank there. So this is actually our most commonly reported taxa at the moment. And so we issued, in September of 23, we issued this plant alert on the species. And so you can see a screenshot of the front and back of this PDF document. You can see some information about the plant, how it broke down per category by gardens ranking there. Some links down on the bottom left. You can see some recommended actions as well that we're recommending for different stakeholder groups to take care of the plant. And then you can see some images in the screenshot on the right and some other information just about the about this species. Notably, on the bottom, there is a series of green buttons there. So if you were to download this, you can share your feedback on what is labeled on this plant alert. So this is the first and only plant alert that the group has come up with so far. We plan to issue other plant alerts as the data demands, but we don't have any other plans at the moment to send any more out. These are also distributed through our newsletter. So if you are interested in receiving these alerts, then you can sign up to receive these newsletters. We only send out about 2 per year. So not too ominous, we won't fill up your inbox. So that's one action step that I would recommend for you to do. Importantly though, if you're on the call today, if you're joining and you are a representative from a botanic garden or arboretum, we'd love to have you participate and contribute your data. So please reach out to us through these email addresses on the bottom and help us to contribute your data. And then finally, we just produced a video on this initiative, which you can find by Googling our name, Public Gardens, as Sentinels Against Invasive Plants. It's now out on YouTube. And that is all I have for the moment. So I think we might have time for questions, perhaps. I'll go back to the slide.
Yes, thank you so much, Kurt. That was super interesting to hear about the work going on at the Morton Arboretum. So we have time for some questions. I see 2 in the Q&A box. First question is, are there any Canadian public gardens that have joined the Sentinel initiative?
Yes, there are a couple of gardens that have contributed data so far, but I suspect that there are others who might still need to or want to join us as well. I think we have 2 or 3 at the moment.
Awesome. Next question. Kudos, admirable collaboration. What might growers need in order to take action on your info on a level playing field?
What might growers need to take action?
Yes, what might growers need in order to take action on your info on a level playing field?
So what growers might need to do, we have some recommended actions here. I kind of went back in my screenshot to look at this plant alert. These are the recommended actions. So these apply across all the different stakeholder groups. So removing trees that are fruit bearing, eradicating spontaneous populations, developing and evaluating cultivars for reduced seed fertility, planting and selling only non-fruit bearing cork trees. I could give another lecture just on the complexity of this particular species, but essentially making sure that they are not fruit-bearing trees is really important. And then increasing garden enrollment and reporting. So I think the most important thing is to take up this information in your planning process as a grower so that you can, so you won't be hit by, regulation when you have a field of trees, for example. I think that would be a really important win. Take this information up early into your planning process.
Great, thank you, Kurt, for that information. If Kurt's email has been put in the chat, for any follow-up questions. Thank you so much for this talk. I'm really looking forward to keeping up with the newsletter and keeping an eye out for any further plant alert. So yes, thank you so much, Kurt, for joining us. Okay, our next talk, we have Michelle Beloskur from the Midwest Invasive Plant Network. She'll be talking about helping you prioritize and manage invasive plants in context of the Midwest Invasive Plant Network. Thank you, Michelle, whenever you're ready.
Having a little trouble getting my video started here. There it goes. All right. All right, if you can let me know that you see my screen okay.
I see the PowerPoint and the like editing
Okay,
field, but not the viewer mode.
Let me try it this way. Does that look better?
I don't think you're currently sharing your screen.
Okay, one more try. My apologies. Okay, this is what I tried to share before. Oh, I think it's just slow. I think I'm being a little impatient. Okay, it's telling me I'm screen sharing. Are you seeing that?
Yes, so your screen sharing, it's just on it's not on the, oh, perfect. Now it's the full screen of the slide, so you're good to go.
Okay.
Thanks, Michelle.
Yeah, thanks for your patience, everyone. I'm really happy to be here today. My goals today are just to give you a little bit of an overview of the Midwest Invasive Plant Network. If you're not familiar with our organization, talk about some invasive plants that are on our radar and kind of how they come upon our radar, and to share some resources that we currently have available, and then a little bit of information about some upcoming projects and resources that will come out of those projects. So our network was formed in 2006, and as we are a network, we serve a large area of the Midwest. We have 9 states as well as the province of Ontario in our service area. And our mission is to reduce the impacts of invasive plants across this Midwest region. And one of the ways we do that is through outreach and information sharing. One thing that we have coming up this year is the Upper Midwest Invasive Species Conference. We hold this conference every other year, and this year it will take place in Duluth, Minnesota. But it is a hybrid event, so you can also attend online if that works better for you. We are currently doing a call for abstracts. So if anyone has a presentation that they'd like to share at this event, please go to umisc.net and check that out. Some other things that I want to let you know are available. We did a 10-part Emerging Midwest Invasives webinar series last year. And all of those presentations are closed captions, and they're available on MIPIN's YouTube channel. You can also link to it from our website, mipin.org/presentations. And we have some new webinars coming up this year. One is actually next week. So if you are interested in learning more about effective management of knotweed, we have 2 wonderful speakers with a lot of experience in this area who are going to be presenting. And you can register for that now. We also have, really excited to share a presentation from the Invasive Species Center that's going to happen the following month. So that'll be March 19th, I believe. And that is going to be talking about successful management of water chestnut. They've done a lot of great work on that, and they want to share what they've learned. And then a little bit later, I think it's going to be May, we are doing a webinar on goat grazing. I know there's a lot of interest in that topic as well. So in addition to these activities, MIPIN also does quite a few publications. These are just a few that are available on our website right now. So if you manage garlic mustard, we have a really great guide to help you kind of prioritize when, whether, if you should continue to manage this plant. It really kind of takes into account, the fact that we can't do everything everywhere all the time. All of us who manage invasives know this. So this is a great way to prioritize and be successful in the work that you do. We have a guide on why should I care about invasive plants, which is really helpful. It gives you talking points for all the different groups that you might be trying to reach with your invasive species messaging, everything from hunters to gardeners to boaters. We've got talking points for all those groups to help you. And then we have our landscape alternatives, both app and brochure. So the brochure is what you're seeing in front of you. That provides native and non-invasive alternatives to a lot of common ornamental woody invasive plants. But we also have an app version that's more extensive. It covers almost 50 different plants and has some information about them, and then again, several alternatives that are either native or certainly non-invasive. And we have our longstanding invasive plant field guide. So another resource that we have that if you haven't checked out, I highly recommend. Our Woody Invasives of the Great Lakes Collaborative website is just a treasure trove of information on 28 different woody invasive plants. It has information on identification, management, how these plants are regulated across the region. Again, these alternatives to these plants. We have a blog that we try to keep up quarterly. And we're always looking for content. If there's something that you think you have to share with us, we would be more than welcome to include that. So what invasive plants are on the horizon and how do we determine that? I'm not going to go over this too much because we just had a really great presentation from Kurt Dreisilker with the Morton Arboretum. We work closely together on this project. But in case you missed his presentation or just maybe to kind of hit a few highlights, this is a really innovative and interesting collaborative project where public garden and arboretum staff contribute information that they have because they have these collections of plants from around the world that they monitor really closely, and they are often the first ones to notice when a plant is escaping cultivation and might be invasive. So this provides a standardized way for gardens to record those observations and share them. Excuse me. And as Kurt said, the database is sort of restricted access just for gardens, but we are fine now that we have enough gardens contributing and enough data in our database, we're able to share some of the trends that are coming out of that project with other stakeholders and interested parties. So as Kurt mentioned, we have our data dashboard, which keep an eye on that because I think it's going to be updated again very soon. We now have 42 gardens. We just had an event last week and we added gardens from both Georgia and South Carolina. So we're really excited to see this project continue to expand. And again, the plant alert is a really helpful guide that shares what gardens are observing about a specific plant that has been reported repeatedly to our database and what actions we recommend to help prevent the further introduction and spread of that plant. So another way we determine what plants should be on our radar is to ask the experts. So we have been doing a survey almost annually. We did one in 2020, 2022, and then last year, where we ask invasive species experts across our 10 state province region what they consider to be early detection plants. So either these aren't in our region yet, but they're knocking on our door, or they're here, but they're still in relatively smaller isolated populations. Either way, we're really concerned about their ability to continue to spread. So we're just finalizing the results of that survey because we did it right at the end of last year. And this is a lot to digest on at a glance here, but I just to give you a little bit of an introduction, we kind of had 12 plants this year that kind of rose to the top of the list. We had over 50 different plants reported by 73 different experts that contributed to this survey. You can kind of see a breakdown of where those reports came from. I know that's probably a little small on your screen, but we got responses from state, provincial representatives, county, multi-county, which a lot of that is from invasive species management area staff, and as well as some regional, university, and tribal and other resources. But I'm going to focus on this slide and talk about the plants that were named by experts from Ontario that contributed to the survey. And I don't think it'll be a surprise to anyone that swallowwort is on this list. We just had that great presentation from Hayden. Yeah, that plant is definitely of concern. Japanese stiltgrass was also named, Japanese chaff flower, calorie pear, which was named by 9 out of our 10 states and provinces in our region. And then I also added water chestnut, which wasn't one of our top 12 that was reported, but I know that it is a plant of concern for Ontario. And again, just want to reiterate, we'll have that webinar next month on March 19th. And this is not on our website yet, but it should be before the end of the month. This is just kind of a summary that you'll be able to download of these 12 different plants. Again, a little bit more information about the survey itself. And it does go into some detail about how our results varied from last year and which plants are of greatest concern for which state or province. So that's just a free download, and hopefully that can help you. You could pull information out of there to share with your audiences. So moving on, talking about some new initiatives that are kicking off. We have a new project funded by the U.S. Forest Service on early detection of invasive plants. And we're focusing on high priority forests, primarily in Michigan, Minnesota, and Wisconsin. But we're hoping that the information that comes out of this project will also be beneficial to the wider region. We are going to be working closely with some participatory science groups, citizen science groups in those states to expand early detection work, working to increase rapid response by doing some educational outreach. We are hoping to survey 30,000 acres. We are also going to be doing some best management practice webinars and developing some customized field guides, which initially will be available to organizations in those 3 states, but I think eventually that's a resource that we're hoping to expand and make available to a wider audience. Another kind of ambitious goal of this project is to try to improve some regulatory consistency across our region, which is a big task, but we are hoping to at least kind of collect and summarize how different states and provinces are regulating different plants and try to share that information across jurisdictional lines, make sure everybody knows what their neighbor is doing, identify some hurdles, and hopefully brainstorm some solutions. Again, trying to kind of harmonize regulations across the region. And there's also some great work that's going to happen primarily in Minnesota to ensure that our programming is reaching diverse and underserved communities as well. So going back again to some available resources, I did want to point out that we MIPN updated our Keep a Lookout flyers this year. We have one on terrestrial plants and one on aquatic. Each features 16 kind of high priority plants for early detection. And you can download those from our website. Again, our survey results will be posted there shortly. You can link to our emerging invasive webinar series from last year there. And we also have a page on the MIPN website devoted to the Public Gardens project that you just heard Kurt talk about. It can be a little difficult to Google that website. So if you're trying to find it and you're not succeeding, just go to mipn.org. You'll find our page, and that will give you all the links you need to get all the information about that Public Gardens project. As we'd love to have more gardens participating and making sure that the information coming out of that project is reaching a wider audience as well. So MIPN also has a few different management resources. One I've already talked about, but I'm also going to mention the MIPN control database. I have a little more information on that on my next slide. We have an early detection webpage, which is where our survey results will be, but it also has fact sheets and other information about all the priority species for our Midwest region. So if you're looking for a good resource on a specific plant, that's a great place to start. And if you haven't a fact sheet or some kind of article and you'd want us to post it on our website, more than happy to do that. Just get in touch with me. And then I mentioned our Woody Invasives of the Great Lakes website as well, which I just can't recommend enough. The control database, I think is kind of an underused tool, but you can search for over 40 different plants, invasive plants, and then you can do a customized search based on your experience level in management, what kind of habitat you're in, what season you're going to be managing in, because sometimes you don't have a choice. You may only have access to a site certain times of the year. So how can you optimize your management given that situation? And how effective do you need this to be immediately and long-term? And you'll get a whole bevy of options, including, not just herbicide, but all kinds of mechanical options as well. And all those techniques have been vetted by our colleagues at the University of Wisconsin. We also have information about site restoration, because that's always our goal, right? Not just to manage the invasives, but to get to a point where we're actually restoring ecosystem health. So we have a list of native plant and seed suppliers you can find. We also have a restoration webinar series. It's a little dated now, it's from 2018, but it's still, I think, a good resource. And we have a couple of guides on landscaping to help prevent secondary invasions and warding off, invasives moving into your property, one designated for homeowners and one more for people in the land management side of things. And then I just wanted to share this screen. I know there's lots of different ways to access these resources, keep up with MIPIN. So we have a quarterly newsletter, we have an email listserv, we're pretty active on all these different social media outlets. And you can always just get in touch with me. And I'm happy to answer any questions or try to get you the resources that you need. So that is my presentation. I'm happy to take any questions. And I think I'm going to leave it on this previous slide, if that's all right, just in case people want to capture any of that information.
Great. Thank you so much, Michelle. Always amazing to hear all the work and initiatives going on at the Midwest Invasive Plant Network. So we have 2 questions. First question, regarding the species named by MIPIN's 2023 survey of Bio-Ontario experts, are those species that are of greatest concern, such as greatest potential impact, for instance, that have been observed at least once, or invasive species that have the greatest abundance? Not sure if that list is based on impact or abundance. And
not in abundance. It's more these are plants that we want to try and do early detection on. So these are plants that are relatively new to the area, or maybe, and they may be like I know we saw in the previous presentation, there was an infestation of black swallowwort. So there may be places where it is well established, but there may be other parts of the state where it hasn't spread yet. And so it's great if we're keeping an eye out and still reporting new occurrences of that plant. So it's more of an early detection list, not this is already widespread.
Great, thanks, Michelle. Next question. Are there any studies or initiatives you're aware of that assess the impact that terrestrial invasive species have on the Great Lakes directly, either through the woody invasives of the Great Lakes Collaborative or other similar efforts? Thanks.
Let me look at that question again. Studies or initiatives that assess the impact. I can't think of anything off the top of my head, but I will look into that. If someone wants to email me that question, I can look into that a little bit more when I have time, because that's a good question.
Thank you. And yes, Michelle's email is in the chat for any further questions. Just wait a couple of seconds in case any other questions come up. All right. Thank you so much, Michelle, for talking with us about the work at Midwest Invasive Plant Network. I'm greatly appreciated and always looking forward to keeping up with the resources you come out with. Much appreciated. Yeah, thank you so much.
Thank you. Great forum.
Okay, so last but not least of this session is a talk by Leonardo Galindo Gonzalez from the Canadian Food Inspection Agency. His talk is titled Using Genome Scanning, Protein Biotyping, and CRISPR Diagnostics to Tackle Identification of Noxious, I don't know if I'm saying that right, and Regulated Plant. Thank you for joining us, Leonardo, whenever you're ready.
Okay, can you see my screen?
Yep, looks good.
Looks good. Okay, awesome. 1 second here. Okay. Great. Okay. Yeah, well, hello, everyone. And thank you for allowing me to share some of the research that we have been doing at the Molecular Identification Research Lab, the Canadian Food Inspection Agency out of Ottawa. So I realized that the title of my talk seems a little complicated, but I really want the take-home message to be that we're working in molecular technologies to make detection of invasive, noxious, and weedy species more accurate, efficient, and sensitive to allow prevention and stopping the introduction of these species. And so one of the main roles of my lab is to generate tools to advance that molecular plant identification to prevent that introduction of these species into the Canadian environment, and also to monitor trade. And so we want to help answer questions like, how do we efficiently detect noxious seeds in seed shipments of agricultural commodities that come to our country or go out from our country? How do we differentiate the species that look exactly the same? Or how do we help people that do not have the expertise to identify some of these species in the field? So I'm just going to look for a pointer here. And so our main challenge, as I was trying to mention, is for using molecular biology techniques is to be able to differentiate those plant species. And there's no universal barcode. So there's no DNA barcode that can apply to any species, so we can differentiate any two species. There's different regions of the genome with different levels of resolution to identify species. And so additionally to this, the method that I need to implement from the molecular perspective is going to depend on the context. So as I was mentioning, do I need to find a trace amount of a contaminant? Do I need to distinguish two things that look the same? Or do I need to identify something or help someone identify something while I don't have all the expertise to do it, right? So to solve these problems, we need to use different techniques that allow us to increase sensitivity, so be able to detect very low amounts of a contaminant, increase accuracy, know what species we are dealing with, and to increase efficiency. So how do we do things faster and maybe implement high throughput processes that allow us to process hundreds or thousands of samples very quickly? And so I will show you working three projects that we've been conducting in the last 2 years in the lab. So one is DNA barcoding, which allows us to increase accuracy. The second one is on protein biotyping, which allows us to increase efficiency. And the last one is an assay based on CRISPR-Cas technology, which is mainly to increase sensitivity. So for this first project here that I want to talk about, it's basically based on the problem that I just mentioned, which is the lack of a universal region in the DNA that allows us to identify any species. And so in this table here, what you can see here is that we have morphological identification for 27 specimens that were sent to us by inspectors from the Canadian Food Inspection Agency. And then these two columns are two regions in the genome of the chloroplast and the nuclear genome that allow us to probably or potentially identify some of these species. And so you will see that in two cases here, there was no identification at the morphology level, but when we look at the respective barcodes, at least one of the two barcodes was able to identify the plant, even if morphological identification was not possible. But in many cases, we also find that a plant that has been morphologically identified cannot be identified with these two regions in the genome, which are very common regions that are used as barcodes. So we see that the main problem here is that there's instances where the DNA barcoding from the available information doesn't allow us to identify plants. And so we need novel regions in the genome that have enough resolution power to distinguish some of our target species, invasive species, weedy species. And since chloroplast genomes happen to have lots of variability when we compare them between closely related species, we use a technique called genome skimming, which allows just to gather a lot of sequencing information from high copy regions in the genome, like the chloroplast, and allows us to assemble full-length chloroplasts. And chloroplasts have 150,000 characters. So lots of information to pick from and try to find new regions to identify plants. And so to test this methodology, we targeted the genus Amaranthus, which contains several species, including Amaranthus tuberculatus, also known as tall water hemp, and Amaranthus palmeri, known as Palmer amaranth. So water hemp seeds are regulated at the federal level in Canada, and Palmer amaranth, even though it's not regulated in Canada, it has been regulated in several states in the US due to the impact that this plant has in several major crops, such as corn, sweet potato, soybean. So Palmer amaranth has been detected in the provinces of Manitoba and Ontario, not as established populations, but it has become a concern to the Canadian authorities now. And so these species can produce, each one of the plants of these species can produce up to 1 million seeds under ideal conditions. And they have multiple herbicide resistance, and the seeds are really difficult to distinguish due to plasticity. And so even for fully grown plants, flowering is required sometimes to distinguish the species. So what we did is we used 28 samples that we have that correspond to 15 different amaranth species, and we assembled, with the technique that I mentioned, we assembled full chloroplasts, 150,000 nucleotides. And so we aligned these chloroplasts, and what I'm showing here is just a small region of those 150,000 nucleotides, which provide different levels of resolution to distinguish between 15 species of amaranths that we have. So what you're going to see here is the name of the species, and these blue dots or lines that you see on your screen correspond to specific nucleotides or characters that are sometimes shared or sometimes unique for each one of the species. But we use 7 different regions from the chloroplast and to distinguish some of these species that we had. And we received some samples from our genotyping and botany lab within the agency. And we were able to identify all those samples with 100% accuracy. We were even able to identify 2 samples for which morphological identification was not possible. So this way of identifying plants is very basic and requires doing sequencing every time. So it takes a little time. It has high accuracy for identification, but it's not something that you would like to do always. Sometimes you have to have a test which is faster. And so some of these polymorphisms or changes can be turned into rapid tests to identify plants faster than just having to sequence. And I'll talk about one of these tests later on. But now I want to speak about the second methodology. We use that sometimes does not have the level of resolution that DNA barcoding might have, but it does provide an efficient high throughput method for identifying plant species. So you can process 100 or 200 samples in a couple of days using this methodology. So this methodology is known as MALDI biotyping or mass spectrometry biotyping. I'm not going to go into the acronym, but it's commonly used to generate protein fingerprints to differentiate microorganisms. However, I found doing some research, I found that there was a group of scientists that was working with this methodology in some invasive plants, and we tweaked the protocol a little bit to use it in plant seeds to see if we could do plant identification from plant seeds or from individual plant seeds. And so identifying species from seeds, as I mentioned, is often difficult because they are so plastic, so they are easily confused sometimes. And so what we basically do with this methodology is to pulverize each one of the seeds, and then we place them, so we extract the protein and we place them in a plate that goes into this equipment. That's the MALDI biotyper here. And then the protein fractions get ionized by shooting at them with a laser, and then they fly through here, depending on their mass, their weight. And when they come out, they create these profiles that you see here. Each one of these peaks correspond to a major protein fraction for that specific species. And so initially we did a test here with some plants from the canola family, just because we had lots of those seeds and we were testing the methodology. And you can see that there's differences in the peaks that are produced between each one of these different species of plants. However, when you compare 2 accessions of the same species, the profiles are basically the same. And so, of course, what we really want to do is to use this technique to target species of concern, and then we use amaranths again. So we generated a database of protein profiles from 16 amaranthus species. I'm not going to tell you about the technical details, but we did a lot of replication, a lot of different accessions per species to account for variability. And then after you do that, you want to process your blind sample. You receive samples that you don't know what the species are for those samples. Process them the same way, and then compare the protein profiles to the protein profiles on the database to see what they match. And then you get a heat table to analyze. And so for the first set of blind samples that we received 15 blind samples that you see on this column, the first column here. And you have the identification call made after comparing the protein profiles by the software of the MALDI biotyper. So you'll see that there's 3 hits per each one of the samples. That's basically because we took 3 replicates or 3 seeds per each one of the vouchers that we received. And then you have an average score of the similarity. Usually over 2 is a good similarity. And on the last column, you'll see the original morphological identification that was revealed to us only after we made the protein identification. So in this case, we had 100% accuracy. There were a couple of cases where one of the 3 replicates did not correspond to the expected species, but 2 out of the 3 did correspond to the expected species. So we use kind of like a majority rule there. So we did a second experiment after this first one that was 100% accurate, and we received 60 blind samples from the Canadian Food Inspection Agency in Saskatoon. And the second experiment was 87% accurate. And so most of the mismatches on the second experiment were due to the fact that we didn't have all the species in our database that they sent us as blind samples. So this work has already been published in Plant Methods. And so for our third method, going back to using those differences in DNA, I want to talk about something that I think is just the coolest stuff that we're doing right now. The last technology I will speak about today is very well known, especially in the molecular biology field. And the people who developed the technology won the Nobel Prize in 2020. And so the idea actually came straight from Jennifer Doudna, who's one of the Nobel Prize winners. While I was listening to her on a CRISPR forum a couple of years back. So CRISPR systems were first discovered in bacteria, and they resemble immune systems, mainly comprised of enzymes which cut foreign nucleic acids from invaders, and those invaders are usually, for bacteria, they're usually viruses. So Doudna and Charpentier repurposed this bacterial system to use it in creating mutations in any organism, in the DNA of any organism. So the way CRISPR-Cas systems work is by generating a complex between something that's called a guide RNA. And this is a molecule that you usually design. It's a single strand RNA that will be complementary to a region in the DNA that you want to mutate. So the complex is created between the single guide RNA, an enzyme, and then your target sequence. And then molecular scissors will cut both strands of the DNA. And after this happens, the host repair mechanisms will try to repair that cut section, but this mechanism is error prone. So every time that the host tries to repair it, it introduces a mutation. So usually this is very commonly used to produce mutations in genes where you want to just corroborate that there's a specific function. Now, for Cas9, the Cas9 is the enzyme that is usually used for mutagenesis, but there's other enzymes in the same family that have additional functions. So for example, here I'm showing Cas12, which does pretty much the same. So you have your enzyme, guide RNA, double-strand DNA, and then your target, which is cut. However, when Cas12 finds a target, it will also cut free-floating DNA strands. So why is this important in molecular diagnostics? Well, if I have a region in DNA that's different between 2 plants, I can design a CRISPR or a guide RNA, which is very specific for one of those polymorphisms. And when Cas finds its target, we can add this single-strand DNA that has a reporter system with a fluorophore and a quencher. So as long as these are together, nothing happens. But when this single-strand DNA sequences are cut, then the fluorescence is released. You can produce fluorescence. So the way that we made this assay for our case is, so we had some plants from Amaranthus extract DNA, amplify the region where the guide RNA might bind, and then do our complex, our enzymatic complex along with our reporter to produce fluorescence. And so we have done many experiments with this, but we basically created a guide RNA, which is specific for Amaranthus palmeri and the sister species, which is called Amaranthus watsoni. And here we tested 70 different species with different accessions, and you only see the acronyms of the species here, but just to say that from 32 to 38 here, These are all Amaranthus palmeri, and 64 is the sister species, Amaranthus watsoni. None of the other species here are showing fluorescence after 1 hour of doing the reaction. So the cool thing about this is that you can actually do amplification and the enzymatic reaction in under an hour without any equipment. You can do it at room temperature, right? And we are tweaking the protocol to make it even more efficient. So our idea is to implement this so we can do it almost for, I mean, any 2 species or any different species that we need to test. So with that, I just want to say that we have a series of different projects in the lab that we're working on, and we'll be having upcoming opportunities for either grad students or postdocs in some of our projects working with wild oat and with different techniques, different molecular techniques. We also have a really neat project that we want to do using pollen as a proxy for detecting plant pathogens and invasive plants. And with that, I want to give my acknowledgement to some of my collaborators, and here's my contact information in case anyone wants to further discuss some of these technologies. Thank you.
Thank you, Leonardo. So interesting to hear about molecular tools in progress that are being implemented. Thank you so much for this talk. As per usual, any questions, please put them in the Q&A box, and we have a few minutes left to answer. Wait a couple, a little bit more for questions to come in. Thank you so much, Leonardo. Further questions can be directed to, any questions can be directed to his email, which we will put in the chat if that's okay with you, Leonardo. Actually, one question's come in asking additional species. Are there any additional species for this work?
Yeah, so right now we are for the CRISPR-Cas, we are testing the technology with a plant pathogen called Phytophthora. And you are also collaborating with the plant pathologist working on oak wilt. And so, I mean, the technologies are highly transferable to almost any species. You just have to, depending on what you want to use, create a database of protein profiles, or just find the polymorphic regions in the DNA to make sure that you can create the test.
Next question. Is the technology available to nonprofits?
Yeah, well, the idea with this is that, I mean, once we have performed all the experiments and we have completed kind of like a core story of what we want to do, we're going to have a publication out. And so all the basic methodology will be published and freely available for whoever wants to use. I mean, the idea in the CFIA is that we also transfer technologies to our diagnostics labs, which is a longer process, but the methodology itself will be published in peer-reviewed journals.
Amazing to hear. Okay, I think there's a couple more questions. I think Maddie would be good to keep going with those. Sounds good. Sorry, I may have, this is in the Q&A box. Someone asked, sorry, I may have missed it, but can CRISPR be applied to any plant part or only seed?
No, it can be applied to anything basically, because you have the same genetic makeup in any cell in the seeds. The only problem is like if you use chloroplast DNA, there might be some plant parts that may not have as much of the DNA, and there's some parasitic plants that have lost their chloroplast or have lost parts of the genome. So in that case, you have to be aware of that. But you can also create the CRISPR test based on nuclear genomes. And so, yeah, I kind of like mumbled there a little bit, but yeah, like any part plant would be suitable for applying CRISPR. Yeah.
Great, thank you. And someone just has a follow-up from their original question from is the technology available to nonprofit? They ask how long away will that be?
Yeah, I guess they're asking how long before we publish. I'm writing a paper right now, so it might take a few months. So the whole process of publication to the actual publication being out for the public sometimes takes anywhere between 3 and 6 months. So I anticipate that at least preliminary work will be published in 6 months.
Great. Well, thank you so much, Leonardo, for your talk. And his email again is in the chat for any follow-up questions. And I think that concludes. Thank you for concluding
Thank you so much.
this session, Leonardo. There's such an amazing variety of talks, and it's always great to hear about CRISPR. It's such an exciting development the last few years. So, and yeah, such an amazing variety of talks. Thank you so much to everyone, all the speakers that came out today to share their time and knowledge. And I'll hand it off to Maddie.
Thanks, Nicole. And yeah, special thanks to our speakers for sharing their knowledge and projects with us here today. We're going to take a short break here and rejoin at 11:45 for invasive species trivia. So quickly refill your coffees or your water and get ready to compete. We're excited to see you